Okay, this is a little video about translation. I've just realised that it's gone off the top instead of the bottom. Oh, never mind. No, I can do it. That's okay. Oh, there's Mr. Sowell's face. Way! Okay, I'm going to be careful not to go off the bottom now. I'll keep an eye on it. Don't worry, it'll be all right. So, I was just saying, it's my least favourite stage of protein synthesis. There's piles of stuff going on here. So transcription, really straightforward. You make that complementary messenger RNA molecule using RNA nucleotides, and it exits through the nuclear pore, which is lovely. Now I'm not going to start with what mRNA does next. I'm going to start up here in this little corner here, because we have other sort of RNA, which is tRNA, and that has at the top end, that has an amino acid binding site, and at the other end it has 3 unpaired bases called an anticodon. And the anticodon is the determining factor. I'm not going to say that it decides anything because it's an anticodon, it doesn't know anything. It's the determining factor for which specific amino acid is carried. But before you can actually carry it, you've got to attach it, and that's going to involve a bit of ATP, so it's an energy consuming process. And this process is called amino acid activation. I don't know why it's not called tRNA activation, but anyway, it's not, so there we go. And so I'm sort of simplifying my tRNA into this sort of, I don't know what you call it, a bow. And it'll end up with its amino acid attached to it. So that has to happen, and wherever you've got free transfer RNA and some ATP, you'll attach amino acids to it. So that's sort of, part one of the story. So anyway, back to messenger RNA. See, I told you there was a lot going on in this. So the messenger RNA comes out of the nuclear pore, and it attaches to a ribosome in between the large and small subunit. Oh, I think I've got room there. In between the large and small subunits. So there we have our ribosome, and you'll see that there's room for 6 nucleotides across there. Now that sort of number is quite significant, because every 3 nucleotides on the transfer RNA is called a codon. So this isn't a big shock, is it? Because we've got 3 nucleotides, 3 free bases in our anticodon, we've got 3 bases sticking out of our codon. So this is a triplet code, and each 3 bases, what that means is 3 bases code for 1 amino acid. And it really is not rocket science, and you should be now thinking, I wonder what the anticodon's got to do with the codon. I wonder if they're complementary, perhaps. And if you're thinking that, I think you've cracked it. So if we have our first codon being AUG, I'm making these up, just like to point that out now, then what will happen is, as these tRNAs carrying their amino acids are sort of colliding around, careering around inside the cytoplasm, they will collide with their complementary anticodon, and they'll be carrying their specific amino acid, which I'm now going to draw as a circle, just, hey, let's mix it up a bit. So here, what we've formed here is, I'll go back to my black pen actually, it's a bit sharper. Is an anticodon, codon complex. And of course there are little hydrogen bonds connecting those, connecting them together, and there's sort of a temporary state of affairs. And here we've got our specific amino acid. Now the reason that we've got room for two is that what a protein is a chain of amino acids. So we might have another code. Again, kind of pretty much going to make this up. So I'm going to go AAU for this one, and hopefully that will remind you that the base in RNA is U rather than A. So this will be our next one, and it will have an amino acid attached to it specific to that anticodon, and we're going to get our hydrogen bonds attaching them together, so we've got another codon-anticodon complex. And the idea now is that these two amino acids are close enough, they're brought into close proximity to be joined by a peptide bond. So the adjacent amino acids joined by a peptide bond. So now we can see that we've got the code from DNA, we've made a complementary copy, we've got complementary anticodons, they're carrying specific amino acids. So the code in the DNA determines the order of the bases in the triplet code on the mRNA, which determine which amino acid is brought down, so that these amino acids are brought down in a joined together in a specific sequence for that protein. So that's sort of, that's quite important really. What happens next is that the ribosome trundles along another to the next codon down. So I'm going to just draw this in a dotted line. So here's my ribosome. It's moved now to position to the next codon. And what will happen then is the next amino acid will come down and occupy position 2. So the next tRNA will bring its amino acid down to position 2, and the same thing will happen again. So what happens to this one? Well, it's kind of dropped off the ribosome, if you like. So here, what happens is that the tRNA, without its amino acid, it's left that behind, attached to that one, returns to pick up a new amino acid. So it'll pick up another one out of the cytoplasm if there's ATP available. So that by the time you get to the end and you get to a release factor, what you've got is your chain polypeptide of amino acids. Remember, primary structure, specific sequence of amino acid, depending on the codons, the order, which determines the order in which those are brought down. That, of course, we would call, so this will be the primary structure, which is the sequence of amino acids. Now clearly there is more to that story, which we did in the core, and the next sort of stage will be to trundle off into the Golgi body and vesicles from the rough endoplasmic reticulum and be modified and folded up into the secondary, tertiary or quaternary structure, or have a short chain of saccharides, oligosaccharides added to them and make a glycoprotein. Whatever's going to happen to them is going to happen to them in the Golgi body. So I'm pretty much hoping that that is the story, and Dr. Saville's gone away so I can't tell. I'm going to leave it there.